In glycolysis, a 6-carbon sugar called glucose is broken down into 2 molecules of 3-carbon pyruvate. This multi-step process produces a net gain of 2 ATP molecules, 2 high-energy electron-carrying NADH molecules, and 2 molecules of water. Overall, glycolysis consists of 10 enzyme-catalyzed reactions.
Glycolysis begins with the phosphorylation of glucose, which prepares the molecule for the reactions that follow. Here, a phosphate group from ATP is transferred to the 6th carbon of glucose, forming glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase.
Next, the enzyme phosphoglucose isomerase catalyzes a reversible rearrangement of glucose 6-phosphate to form fructose 6-phosphate. This reaction changes the structure of the molecule without adding or removing any atoms.
Then, fructose 6-phosphate is phosphorylated at the 1st carbon using ATP to form fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase.
After that, fructose 1,6-bisphosphate is cleaved between the 3rd and 4th carbon, producing 2 3-carbon molecules. These are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The reaction is catalyzed by the enzyme aldolase. From this point onward, glycolysis proceeds using 3-carbon molecules.
Next, dihydroxyacetone phosphate is reversibly isomerized into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. Although the reaction is reversible, glyceraldehyde 3-phosphate is continuously used in subsequent steps, driving the reaction in that direction. This step completes the first phase of glycolysis, resulting in 2 glyceraldehyde 3-phosphate molecules.
Then, both glyceraldehyde 3-phosphate molecules are oxidized. In this step, an inorganic phosphate is added to glyceraldehyde 3-phosphate, forming 1,3-bisphosphoglycerate, which contains 2 high-energy phosphate groups. At the same time, NAD+ accepts hydrogen ions and is reduced to NADH, conserving energy for later use. The reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase.
Then, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate as a high-energy phosphate group is transferred to ADP, forming ATP. This reaction is catalyzed by the enzyme phosphoglycerate kinase. Since 2 molecules of 1,3-bisphosphoglycerate are formed from 1 glucose molecule, this step generates 2 ATP molecules, making it the first ATP-producing step of glycolysis.
After that, 3-phosphoglycerate is rearranged to form 2-phosphoglycerate. This involves a simple internal shift of the phosphate group from the 3rd carbon to the 2nd carbon. This reversible isomerization reaction is catalyzed by the enzyme phosphoglycerate mutase.
Next, 2-phosphoglycerate undergoes dehydration to form phosphoenolpyruvate by releasing 2 water molecules. This reaction is catalyzed by the enzyme enolase. Finally, phosphoenolpyruvate transfers its phosphate group to ADP, producing ATP and pyruvate. The reaction is catalyzed by the enzyme pyruvate kinase, and this step completes the process of glycolysis.